An antibody or immunoglobulin is an antigen-binding protein, produced by the body's immune system. When our immune system encounters an antigen such as toxins or pathogens, B cells get activated immediately. These activated B cells are called plasma cells, that secrete enormous amounts of antibodies specific to a particular antigen. In general, antibodies are identical to B cell receptors, except for the C-terminus region. In the case of B cell receptors, the C-terminus is a hydrophobic membrane anchoring sequence, and in the case of antibodies, it is a hydrophilic sequence that allows secretion. An antibody is a Y-shaped structure, comprising of three equal-sized globular arms which are joined by a flexible stretch of polypeptide chain known as the hinge region. A typical antibody is made up of 4 polypeptide subunits. They are 2 identical heavy chains, and 2 identical light chains. The 2 heavy chains are linked to each other by disulfide bonds. Similarly, each heavy chain is linked to a light chain also by a disulfide bond. Light chain consists of polypeptides of about 25 kilodaltons, and the heavy chain consists of larger polypeptides of around 50 kilodaltons. Altogether, a typical antibody is an 150 kilodalton globular protein. An antibody is made up of a variable region and a constant region. The region that changes to various structures depending on differences in antigens is called the variable region. This region consists of 100 to 110 amino acids, and it differs from one antibody to another antibody depending on antigen structure. The region that has a constant structure is called the constant region, and this region exhibits limited variation. An antibody has 2 main parts which are involved in its functions. They are fragment antigen-binding or Fab fragment, and fragment crystallizable region, that is, FC region. Fab fragment is the N-terminus region on an antibody that binds to an antigen. This antigen binding site is also called the paratope, as it specifically binds with the epitope of an antigen. A typical antibody has 2 identical Fab regions, and therefore binds to 2 identical antigens. It is composed of 1 variable and 1 constant domain of each of the heavy and the light chain. Here, the variable domains are responsible for the antigen specificity of an antibody. The FC region is the C-terminus region of an antibody that interacts with cell surface receptors called FC receptors. The interaction of the antibody with an effector cell activates the immune system. Antibody classification is based on unique amino acid sequences in the heavy chain constant region that confer class-specific structural and functional properties. The different classes or isotypes of antibodies are IgG, IgA, IgM, IgD, and IgE, with gamma, alpha, mu, delta, and epsilon heavy chain types respectively. IgG is the most abundant antibody class. It constitutes about 80% of total serum immunoglobulin. It is present on the surface of mature B cells and in serum. There are 4 subclasses of IgG. They are IgG1, IgG2, IgG3, and IgG4. IgG is the only antibody to cross the placenta, and consequently, it transfers passive immunity from mother to fetus. Newborns therefore have high IgG concentrations in the first 3 to 6 months. IgA is the most prevalent antibody in secretions, such as breast milk, tears, saliva, and mucus. It constitutes about 10 to 15% of total serum antibodies. There are 2 subclasses of IgA, they are IgA1 and IgA2. IgA forms a dimer, where a joining chain, or J chain, connects 2 Y-shaped molecules, giving it 4 antigen-binding sites in total. Pathogens bind to the epithelial cells through specific receptors. IgA antibody binds to the surface of pathogens and neutralizes them so that they cannot bind to their receptors. These antibody-antigen complexes are easily entrapped in mucus, and are eliminated by the ciliated epithelial cells of the respiratory tract. IgA is therefore the first line of defense for mucosal surfaces such as the intestines, nose, and lungs. IgM antibodies are expressed on the surface of B cells as monomers, but secreted as pentamers. It constitutes about 5 to 10% of total serum antibodies. The pentamer has 5 antibodies connected by a J chain, with 10 antigen-binding sites in total. It is the first antibody produced during fetal development, and the first to be produced by B cells against a new infection. IgM is involved in agglutination and opsonization. IgD is present on the surface of B cells. It constitutes about 0.2% of the total antibodies in the serum. It acts as a receptor on the B cell surface, and participates in B cell activation and differentiation. IgE is mainly found on mast cells. It is present at very low concentrations in the blood and extracellular fluid. It is associated with allergic reactions. IgE binds to allergens, and this complex is recognized by mast cells. This binding triggers mast cells to release histamine, a process called degranulation, which is generally an allergic response. This process eliminates allergens. IgE is also part of the body's response to parasitic infections. These are types based on the heavy chain isotype. Based on the light chain, there are 2 types, they are kappa and lambda. Each antibody can have either 2 kappa or 2 lambda chains, but not one of each. That's all about antibody structure and classes. Please subscribe and follow us for more information on antibodies.